Okay, so after that very brief break, I want to take us into our final session of the 2021 Nonprofit Forum. I want to make a note about a change in our agenda. Our executive committee member who was originally going to moderate this session, Susan Dickinson, unfortunately was pulled away today, but we have another executive committee member, Janet Heischetter, who has graciously stepped in. And so what you are going to see now is actually the pre-recorded introduction by Susan, leading into our presentations by our speakers, Allyson Berent, Nathalie Maitre, and Dominique Pichard. And then we will shift into our live Q&A session that will be led by Janet. And we are really grateful to have such a fantastic executive committee that really helps us plan this entire agenda and is able to really pitch in when things come up. As we know, that is how life goes these days, particularly when we're all trapped on the computer. So without any further ado, we are going to play our pre-recorded introduction and presentations on success stories and the importance of early diagnosis.
Good afternoon and good morning to those of you joining us from the West Coast. My name is Susan Dickinson. I'm CEO of the Association for Frontotemporal Degeneration. I'm a member of the NINDS Advisory Council and also a member of the Planning Committee for this nonprofit forum. It's my honor to moderate the final session of this year's forum on success stories, the importance of early diagnosis. I really enjoy moderating this session because while we all love hearing about advances and breakthroughs in other diseases, I know I personally always want to understand how it happened. What did that leader or group do that laid the groundwork for that success? Maybe they framed the question in a certain way. Maybe they focused all of their resources on one specific question that opened up the field. Maybe they brought in guidance from a different, distinct perspective. Maybe they were able to find common ground across a number of funding organizations. And of course, there's always the X factor we call luck. But I'm a firm believer that smart strategy can position you to take advantage of luck, even if you can't control when or where it happens. So our goal today will be to listen to success stories from 3 advocate scientists, and then together try to identify some nuggets of truth, something the rest of us can learn and hopefully apply in our own work. So joining us today are Natalie Maytree, Allyson Berent, and Dominique Pichard. Natalie is an MD PhD board certified neonatologist who's also a research investigator with a focus on neurodevelopment in high-risk newborns and rehabilitation of long-term disabilities. She's the director of the NICU follow-up program and the medical director of NICU developmental therapies at Nationwide Children's Hospital. She's going to speak today on her work transforming early detection and intervention for cerebral palsy. Dr. Allyson Berent is a veterinary internal medicine specialist who serves as the director of interventional endoscopy services at the Animal Medical Center in New York City. Dr. Berent is also a member of the board of directors and chief scientific officer for the Foundation for Angelman Syndrome Therapeutics. She's going to be speaking today about her work stimulating and enabling translational research opportunities that have brought novel genetic therapies forward into human clinical trials for Angelman syndrome. And our final speaker today will be Dominique Pichard. Dominique is an MD who's focused her career on patients with rare disorders and complex medical needs. Prior to joining the International Rett Syndrome Foundation, Dr. Pichard held a joint appointment in the pediatric oncology branch and the dermatology branch at NIH, where she worked as a clinical investigator. In addition to caring for patients with complex medical needs, her research focused on identifying new treatments and improving clinical trial design for rare diseases. Dr. Pichard is going to be speaking to us today about her work as Chief Scientific Officer for the International Rett Syndrome Foundation. And so we will get started, and I will ask Dr. Maytree to please share her slides with us.
Thank you, Susan, for that eloquent introduction, and thanks to the NINDS for inviting us to speak at this incredible forum. As Susan said, I'm Natalie Maytree. I am a physician, and I care for babies and young children who are at the highest risk for a disorder called cerebral palsy, or CP. I'm a neuroscience researcher, and I spend my time designing new interventions and clinical pathways for infants who've had perinatal brain injury. I'm also the mother of 2 boys. One of them was born preterm and now has cerebral palsy himself. I'm going to speak to you about transformation of the early detection and intervention process for cerebral palsy, at least in the US. So the problem is this: for cerebral palsy, which is one of the most common physical disabilities in the world and in the US, best evidence exists on how to diagnose it early, but clinicians don't use it in practice. This evidence was created through systematic reviews of multiple research over tens of years, and a group of multidisciplinary stakeholders from the science, medical, and community worlds got together, and they all made a list of guidelines based on this best evidence on how people should detect cerebral palsy early and diagnose it early so that early interventions could happen. So why then, since these guidelines exist, were they not in practice? Because all the elements of these guidelines were actually present for the last almost 20 years. So once we knew what the problem was, we then also had to look at the consequence of it. Because in clinical practice, cerebral palsy is not diagnosed until after the age of 2, usually, researchers can't develop new interventions as early as possible when the brain is most plastic and when parents and children could benefit most from interventions that support them and their children. Often they have to wait years. If this were a stroke in an adult and someone told you, you've just had a stroke and you're going to have to wait 2 years, and then we will give you an intervention that will treat this, how would you feel? So we wanted to address this problem, and the solution for it was to implement these guidelines for early detection and diagnosis for cerebral palsy. This was done through 3 main processes. First, a really well-aligned partnership with the Cerebral Palsy Foundation, which is a small foundation, not one of the big research foundations, but one whose mission was dissemination and knowledge translation, which was really unique among the cerebral palsy foundations that existed. Then the use of implementation science, which is not your typical science, but it's a very practical one, and of models that allowed us to design single tools and implement them and scale them up first locally and then nationally, and then to start working with state collaboratives. And then finally was to use for our success measurement metrics that healthcare systems would actually care about, but also not forgetting that we're doing this for parents and families and researchers so that researchers can help design new interventions. When we implemented the guidelines that I spoke to you, these international guidelines for best evidence, the results were pretty stunning. Across a network of 5 institutions in the US that represented about 11% of babies being diagnosed who are at high risk for these diagnoses, we decreased the weighted average age at diagnosis from about 19.5 months, near 2 years, to 9.5 months. CA just means corrected age. A lot of these babies are preterm. So fine, that was a great success. But the other thing that we showed for the healthcare system is that the way we did this was by increasing the number of very targeted visits at 3 months with certain tools and elements, standardized testing and personnel in them. And we also did US level parent feedback on the ways of coping they had with these new diagnoses and what they saw as necessary improvements in the process. All of this allowed new interventions to be put into the NIH for new research studies, which some of them actually just got funded. So the lessons learned from what we did was that the reason we think our team was successful in implementing early diagnosis for cerebral palsy in the US is that we found really well-aligned partners, foundation partners, but also partners at hospitals and clinics and parents. And these partners all believed what we believed, and they believed in the why of what we did. We also took these enormous big vision plans and implementation of an entire set of guidelines. You know, there's a myth in implementation science that it always takes 17 years to get new research into practice. And we said, we can do this in 1 year if we just break it into achievable elements that will make them more manageable and will get us more buy-in, and we will leverage really well-studied methodologies instead of trying to recreate new ones. And finally, we had deliverables that multiple stakeholders valued, not just our parents and families, not just our researchers, but we knew that the healthcare systems as a whole would value the process metrics and what I didn't show you, the balancing measures that we studied. So we started with the golden circle, which is why, how, and what. The most important thing being why we did. And so I will tell you that the obstacles we faced and that we had to overcome were mostly misunderstandings in communication that the why often helped overcome. Thank you so much. And it's going to be Allyson's turn now, and I'm going to hand it over to her.
Great. That was a wonderful talk, Natalie. Thank you so much for sharing your story. And I hope to share a little bit of mine as well. I would also like to thank the NIH and the NINDS for inviting us to be here today. It's really a great honor to share some of our success stories and be even recognized to be part of such a great panel. So thank you for having me. I'll start also by just quickly introducing myself. Some parts that weren't in my bio is that, yes, I am the Chief Science Officer for the Foundation for Angelman Syndrome Therapeutics, and I am the Chief Operating Officer of a biotech company called Genetics Biotherapeutics that is singularly focused on the development of an antisense oligonucleotide for the treatment of Angelman Syndrome. I also serve as the director of the Angelman Syndrome Biomarker and Outcome Measure Consortium, as well as the co-director of INSYNC, the International Angelman Syndrome Research Council. I am a veterinarian by training and during the day, and I do clinical research in animals with naturally occurring diseases through medical device development. But the most important job that I have is I'm the mother of this beautiful little girl named Quincy, who's 7 years old, and she lives with Angelman syndrome. And so I'm going to talk about our success, and I think it's really important to walk you through a little bit of the journey to get there, because I think the only way you can really learn from success if you understand the parts that make it successful. And so hopefully in this short stint of time, I can share with you a little bit about how I learned when I got the diagnosis of Angelman syndrome and how that really changed the trajectory of how this disorder particularly was approached and treated. And so the first thing I did was I had to understand what is Angelman syndrome and the neurogenetics behind it. And so for me, that was not as difficult as it is for many, because I am a clinician. And most of us on this call are, but not everyone starting foundations and working in rare disease is. And so knowledge is power, and really understanding the biology behind different disorders is important. And Angelman syndrome has a very unique biology. Its epigenetics is very interesting, and quite the interest is what has made so many pharmaceutical companies interested in developing therapeutics for it. So it's an imprinted disorder, and individuals are missing a gene called UBE3A, but there's a silent copy of that gene sitting on one of the alleles, and it's a disorder only of neurons. So that's really important in understanding how to approach the disorder. And so by understanding that, what I realized very quickly after my daughter's diagnosis at 5 months of age was that it is a rare non-degenerative disorder that is monogenic, and it is neurodevelopmental. And so ultimately what happens with any individual with Angelman is they have quite a homogeneous list of symptoms. And that is also very important when you're thinking about clinical trials and how to design them. So individuals with Angelman have a universal lack of speech. They have life-threatening and debilitating seizures at times. They have severe developmental delay. They have motor dysfunction. They are incoordinated with significant ataxia. They have dyspraxia, as well as some interesting behavior characteristics, most of which are a very happy demeanor. They do have sleep disturbances. Some kids will sleep 2 or 3 hours a night. And the most important thing about Angelman is that these individuals are unable to live an independent life. And there's a significant clinical unmet need and not an approved drug that's currently available for Angelman. And so quickly after our diagnosis, I was introduced to the largest non-governmental research funding foundation in the world for Angelman called FAST, the Foundation for Angelman Syndrome Therapeutics. And within a year of her diagnosis, I was so humbled to be asked to join the foundation as a member of the board of directors, and quickly wound up becoming their chief science officer. As a translational researcher myself in animals, ultimately what I was able to appreciate very early on, that there was an amazing amount of work being done funding research in Angelman syndrome. FAST alone, not counting the NIH and other foundations, have funded over $20 million in research directed intricately toward research for translational treatments for the disorder. And so what I realized very early on in her diagnosis was that a lot of this research hadn't been translated. It was stuck in an academic lab, and there was this valley of death of not knowing how to get it into the clinic. There were no pharmaceutical companies that were interested, and the neuroscientists were doing amazing work, but they really didn't have the expertise or knowledge base and experience to know how to translate that into the clinic. And the amazing clinicians we had working in Angelman were not MD PhDs, and they didn't know how to take it out of the academic lab and bring it to the clinic and understand how to get to human clinical candidate. And so when I joined the foundation, I was tasked with a really great mission, which was the translational research mission. And so what I learned quickly was that the foundation had funded over $20 million of research. $3 million had been directed toward developing novel animal models. There's a pig model of Angelman syndrome, a rat model of Angelman syndrome, as well as 4 mouse models of Angelman syndrome, and now there are 7. There were over 20 laboratories funded by FAST to understand the etiology of Angelman syndrome and understand the different genotypes and phenotypes associated with the disease. And they were really focused on developing different treatment strategies for Angelman. And now at this point, we have over 9 gene therapy disease modification strategies that we are fully supporting in order to get them through to drug development programs. So FAST was launched in 2008, and I didn't join the organization until 2015. So a lot had happened prior to my joining the organization. And what I was able to do was to try to take this collaborative initiative. They created this collaborative consortium of scientists that worked together from 4 laboratories that had weekly phone calls to really get out of their silo and work together in order to create and support each other through their different therapeutic platforms and strategies. It was really an accelerator for bringing therapeutic platforms forward for Angelman syndrome. And so when I jumped in, I was able to take all of this work that had been done and really help to leverage that and shepherd that through. And then in 2017, the foundation launched a for-profit company called Genetics Biotherapeutics out of the not-for-profit, out of the academic work that had been done through this FIRE consortium. And we were able to take that forward through a company, through investors and partnerships with a pharmaceutical company called Ultragenyx Pharmaceuticals, and launch a phase 1/2 clinical trial in February of 2020. And so really what is really important in my mind is to create a roadmap for success. And for us, that roadmap was understanding what's needed, because our mission was therapeutic translation. And so we needed to investigate therapeutic strategies by understanding the biology. And for us, that was gene replacement therapies or protein replacement therapies, as well as paternal gene activation. We wanted to identify symptomatic downstream targets in order to understand what the gene does, and then how do we address the targets, because this is a synaptopathy. So how can we help the synapse and have some symptomatic relief of the downstream targets? And then a really important strategy was also to prepare for clinical trials. If we're working really hard to develop drugs for therapeutic translation, we need to understand how to develop, how to characterize these animal models, how to understand what phenotypes are going to be addressed, and how to really understand how to develop clinical trial design that is going to capture endpoints that are appropriate for this population. So we developed the Angelman Syndrome Biomarker and Outcome Measure Consortium in order to bring forward a pre-competitive space for endpoint development that would have FDA support in terms of endpoints, as well as pharmaceutical and industry support for what would be most important for clinical trial execution. And so by understanding the strategies that we were looking at, we were able to really bring in various different teams in order to help support these programs. And so we have gene replacement therapy, and we have various partners, many of which are in industry. We have paternal gene activation, looking at 4 different platforms, and we have various different industry partners that are working on those platforms and those downstream targets. And so by being organized and understanding that roadmap and really leveraging all of the research and shepherding that through, we are able to really hand off a lot of these programs on a silver platter to pharmaceutical companies who don't have to do a ton of the work because we're doing a lot of the R&D for them. And that has been very, very important in our success. And so as we have that success and we're bringing all these programs forward, it's really important that we help prepare for clinical trials. So we wanted to ensure that we had a disease concept model so that we can understand the burden of disease and the clinical unmet need of the disease. We funded a natural history study through the NIH over an 8-year longitudinal period from 2006 to 2014. And we're leveraging all the data from that natural history study. And now we have another study that's ongoing. As I said earlier, we developed this Angelman Syndrome biomarker and outcome measure consortium, which was very important in understanding all the focus domains of the clinical symptoms of Angelman and how to develop endpoints that can capture those domains and capture the most meaningful and minimal changes that are important to families. It's a very pre-competitive space, and there are over 15 pharmaceutical companies that are invested in this strategy. We have a global Angelman Syndrome registry that takes Angelman Syndrome patients around the world and can compile them in one registry format. And we developed an international Angelman Syndrome Research Council, a brain trust of minds that are not only working in Angelman Syndrome, but more likely working in other disorders where they can bring their expertise to the table in Angelman Syndrome and help us ensure that we do a gap analysis and we leave no stones unturned in order to make sure that all research strategies and all therapeutic programs are investigated. And then, of course, developing models for all genotypes of Angelman. There are 5 genotypes of Angelman syndrome, and it is our mission to not leave any genotype uninvestigated, and that every child and adult living with Angelman syndrome can see a meaningful therapeutic in their lifetime. And so we really need to think about targeting every therapeutic and how that can benefit each patient in different ways. We understand there are different genotypes and different ages that may benefit from different therapeutic platforms. And we never want to put all our eggs in one basket. So we want an ultimate symptomatic cure, and that may be a combination of therapies. We needed to develop very strong natural history data and endpoints that are sensitive to this specific disorder. Otherwise, clinical trial failures are not because of a failed drug, but because of failed endpoints. And that's very important to understand that distinction. So when I think about our 3 greatest successes and all of the work it took to get where we are at this point, and we have a long way to go, the 3 things I think that are most important for us was that we created a very focused roadmap to a cure, which led us to creating genetics biotherapeutics and other potential focused programs that were under consideration at the moment, and both the scientific and a strategic roadmap in order to get to where we want to be, which is the mission to develop therapeutics for all individuals living with Angelman syndrome. We grew a robust Angelman syndrome infrastructure program, which was basically internal R&D, so that we can use expertise in neurobehavior, molecular biology, epigenetics, and help train the next generation of scientists through a program at UC Davis. And so essentially it's an internal CRO where industry partners can go to UC Davis, they can give them their drug construct, they can give them their candidate program, their candidate drugs and their programs, and they can test them with the most expertise in neurobehavior assessment and really understand what those drugs are doing rather than trying to create their IND within their own infrastructure in their own pharmaceutical company. And then finally, the development of the Angelman Syndrome Biomarker and Outcome Measure Consortium, the ABOM, which is incredibly important. We went from one pharmaceutical company at the table right after my daughter was diagnosed to now we have 23 pharmaceutical companies that are sitting at our table. And we have used natural history data, which is robust and longitudinal, in order to really understand the symptoms of Angelman, where they're captured and where they're not, and what is minimal meaningful change, so that we can help develop our own endpoints if necessary, or modify currently existing endpoints in order to be able to utilize those in a pre-competitive way and be able to pass those off to industry to make sure that they can use them in the best way for their clinical trial designs. So we fund that as a foundation rather than relying on industry to fund that, which keeps it pre-competitive and keeps it available for everybody to use. So it encourages collaboration and priorities through significant regulatory support, so we can make sure that no drug fails because of poor endpoints, but only if the drug doesn't work. And that's our job as a foundation to ensure that that happens. So I'm really happy to share all of our successes and happy to entertain any questions as we get to the question and answer session. So now I'd like to pass this off to Dominique, who's going to speak about Rett syndrome and her wonderful experiences there.
Allyson, thank you for passing it over. And I want to start off by thanking the organizers of the NINDS nonprofit forum for inviting me to be part of this excellent panel, and also to my co-panelists for really providing such wonderful talks that we can all learn from the different experiences. So I am Dominique Pichard, and I'm the Chief Science Officer of the International Rett Syndrome Foundation. But importantly, I'm also the mother to a 14-year-old young lady who lives with Rett syndrome. She is what drives my passion for this work that I'm going to tell you about. So first, I want to just touch on what is Rett syndrome. Rett syndrome is a rare neurodevelopmental disorder. It mainly affects females, although there are males that are reported. It is what we call a monogenetic disorder. So it's caused by a mutation on a single gene, the MECP2 or MECP2 gene, which lives on the X chromosome, which explains why it affects females more than it does affect males. This is an early childhood onset disorder, but children that have Rett syndrome have what appears to be normal development for the first 6 to 18 months of life, and then they start to lose skills. They lose all purposeful use of their hands. They lose all of their verbal language and communication. And they also have other motor losses, many losing the ability to walk if they had gained it, to crawl, sometimes even difficulty holding themselves upright independently. But this is not a degenerative disorder. And that was really a key scientific finding, that while there is this initial loss of skills, there's then a stabilization. Now, with really significant improvement in the standard of care, longevity is something that we know about Rett syndrome, where individuals with Rett syndrome can live into their 50s and 60s. But much like Allyson described with Angelman syndrome, these individuals require one-to-one adult assistance for every aspect of their life. I touched on some of how there are many medical comorbidities that go along with Rett syndrome, and many different losses that these individuals have. Sleep disturbance, as Allyson talked about, in Angelman syndrome is very common in Rett syndrome as well. 80% of our individuals with Rett syndrome have seizures. The majority have breathing abnormalities, anxiety, a lot of gastrointestinal issues. Individuals with Rett syndrome commonly have scoliosis requiring fusions of their spine, as well as more easily breaking their bones. And as I mentioned, they're nonverbal, but they also have apraxia, making other forms of communication challenging for individuals with Rett syndrome as well. So this is really quite a complex disorder with a lot of heterogeneity, lots of different systems affected, all due to mutations on one single gene. And so to tell you a little bit about our foundation and what it is that we have done, the International Rett Syndrome Foundation was actually founded in 2007 as a result of a merger of two other foundations, the International Rett Syndrome Association and the Rett Syndrome Research Foundation. Now, the International Rett Syndrome Association was started in 1983 by a small group of very committed and passionate parents. Later, the Rett Syndrome Research Foundation was also started by a group of very committed, very passionate parents. These are parents that wanted to help support other individuals who have Rett syndrome, to make a connection point to other families, which was the initial mission of the International Rett Syndrome Association, as well as to identify treatments for those that are living with Rett syndrome. Taking these two foundations into our current existing foundation, the International Rett Syndrome Foundation, we continue to have a dual-pronged mission. Our mission is to accelerate research to treat and cure Rett syndrome while empowering families with information, knowledge, and connectivity to help those individuals today, while we don't have treatments, to live the best life that they can with their loved one with Rett syndrome. And this is just to show what the foundations have been able to accomplish over the years. Our goal is to create a world without Rett syndrome. And these are many of the impact that we have been able to have, from funding the discovery of the gene that causes Rett syndrome, all the way to helping to fund clinical trials. Now, this timeline is really just to show this doesn't happen overnight. So Rett syndrome was first described clinically in 1966. Diagnostic criteria was first developed in 1984. The gene was identified in 1999. And it wasn't until 2010 that we had our first large clinical trial. It was single center, but it was our first attempt to really demonstrate that this community could participate in clinical trials and that this was a disorder that really would benefit from therapeutics. It took almost 50 years to get there. Now, this timeline, hopefully today, with the technology that we have, with the connection that we have with each other through groups like the NINDS Nonprofit Forum, to be able to connect with other foundations and to learn from each other, to shorten this timeline. And here we are today. Back in 2019, for the first time ever, we had 4 different pharmaceutical companies sponsored clinical trials. Currently, there are more than 40 Rett syndrome specialty centers in the world, and 19 of them are in the US. Now, this has been growing with time, and the investments that were made by our clinicians and by our foundations to be able to continue to get the word out about Rett syndrome has led us to this place where there are specialty centers, individuals that really intimately know how to care for an individual with Rett syndrome around the world. Allyson had talked about a natural history study that was NIH funded, and Rett syndrome was part of actually the same natural history study that Angelman syndrome was part of. But for Rett syndrome, we've had 15 years of funding from the NIH, the Rare Disease Consortium Research Network. And what did this do? This really changed the landscape for Rett syndrome. It brought up what are the main medical issues? What do we need to be doing to screen individuals with Rett syndrome to make sure that they're getting the best care that they can get? We learned what are ways to treat different aspects of Rett syndrome. We learned about what does Rett syndrome look like across the lifespan. And the reason that this was so key and so fundamental for Rett syndrome was not only did it change the care of individuals with Rett syndrome such that we now have longevity in Rett syndrome and is no longer a disorder considered to be fatal in early 20s, but rather individuals living into their 50s and 60s. But as well, because we now understand the longitudinal course of this disorder, it's really helpful for pharmaceutical companies to know what would be expected if there were no treatments and how does my particular drug change the course of this disease. Similar to what Allyson shared in the past, where we had very little, previously no involvement with pharmaceutical companies, you see here in about 8 years ago, we had one pharmaceutical company that was financially investing in Rett syndrome. And this company decided to enter the Rett syndrome space because of our foundation. We reached out to this company. We said, we think that you have a drug that would mechanistically make sense in Rett syndrome. And here are all the ways that we can help you bring this through. De-risk the process. How can we help with that R&D component of what pharmaceutical companies have to do to make that investment feel a little bit less scary, less of a leap? And I have to say that this initial investment was really very critical because it not only helped our scientific and clinical community feel much more comfortable with clinical trials in Rett syndrome, it also opened up the door for other pharmaceutical companies to look at Rett syndrome. So now we have over 20 companies that have publicly announced that they have Rett syndrome programs. And this could be still in early preclinical development all the way through to companies that are actively in clinical trials at this time. So this is a really exciting place to be. We know that no one treatment will help everyone with Rett syndrome. And so we know that we need to have as many different approaches in order to ensure that all individuals with Rett syndrome will have a treatment that will help improve their life. And it takes multiple companies to invest in a disorder to be able to bring all these different therapeutics forward. And so I'll wrap up with just some take-home points of what we have learned and what has made our success possible. Passion drives this work. It was a passion of those original families that started our two legacy foundations, and the passion of the individuals that continue to work in the International Rett Syndrome that help to make a difference every single day. A small yet ambitious group can really make a significant impact. As you'll see in many of the nonprofit foundations, they're quite small. While I say that we're a small nonprofit, the International Rett Syndrome Foundation has 9 full-time staff. For some of you, that may seem big to have full-time staff, to have 9 individuals who really dedicate all of their time to our mission. This started as a much smaller effort and has grown with time. Learning from each other. So much of our course has been from learning from others that have done things before us. You learn from the groups that seem like they are further along. You learn from the groups that are in parallel to you because maybe they're doing things differently that might help you in your process. Don't be afraid to reach out. Don't be afraid to reach out to a doctor that you think might be able to help advance the work for your loved one's syndrome. Don't be afraid to reach out to pharmaceutical companies that you think might have a drug that could work for your loved one's disorder. Don't be afraid to reach out to those of us in the nonprofit space. Again, learn from us, take from us. We all are in this together. We all want to make a difference in the lives of those that are living with these disorders. And we do much more and can make faster advances if we work on this together. Involve biopharmaceutical companies early. The more you know about your disorder, the easier it is to get pharma involved. Allyson really touched on some key aspects, not only the natural history study, but also that clinical trial readiness piece, talking about the outcome measures so that when drugs are tested, you have something to actually demonstrate that the drug is working. But don't be afraid to reach out to those biopharmaceutical companies early. Make those connections. Start those early conversations because they may get you to a place of a clinical trial. And finally, I just wanted to put up my email. I want you to know that I have learned from so many people in this space that I am always happy to share what I have learned and help walk this journey with each of you. Thank you very much.
That was terrific. As Sam said, I'm Janet Heischetter. I'm the Executive Director of the Dystonia Medical Research Foundation, and I'm pleased to be your substitute moderator. I know Susan Dickinson wanted to be here to moderate the Q&A, but we will relay how smoothly it went to her after the program. I want to thank each of you for these informative and really inspiring presentations. It's remarkable what you can do, as you talked about, Dominique, with that passion. And I think that you all have a science background, which is wonderful. And being parents of children, you have that passion that's driving you. I think the common thread through all patient advocacy groups is that passion, right? We can help everybody understand that people are waiting for answers and really drive home that there's work to be done. So I want to also congratulate all of you on the great work that you've done and the success that you've had. We talked a little bit about this morning and in previous forums about the importance of natural history studies. So it was really wonderful to have you highlight the importance of those as well. And earlier, Dr. Bent from the FDA emphasized the need to reach out to the FDA early and talk about what you're capturing in your natural history studies. I wondered if you would just talk briefly about how your natural history studies were developed, and did you reach out to the FDA before you started them, or did you have to sort of reach out later and then do a mid-course correction? Allyson, you want to start?
Sure. So the natural history study started, there's been two. So one that was in collaboration with Prader-Willi syndrome and Rett syndrome. So Dominique, we probably can share a bit of this history, which you may actually, having a 14-year-old daughter, know more about the history where it started than I do. When I came in, it was at the end of the 8-year longitudinal study, and then they were starting a new one. And they got actually the funding from the NIH to do both of the natural history studies. And so there was definitely a collaboration with the NIH, which certainly had the liaison to the agency about the best way to perform that. But definitely over time, our natural history study has significantly been modified. And most of that is because as the biomarker and outcome measure consortium has really dove into what are meaningful and appropriate endpoints for this disease, which most of the neuropsychological tests that are done in natural history studies are not really appropriate for most of these significant neurodevelopmental disorders. And so we have a lot of data on endpoints that are not necessarily the most appropriate, where our kids score so far below the floor or really above the ceiling in that they're too juvenile for our kids in some ways, or they're way too advanced for our kids in other ways. So we have leveraged a lot of the learnings of where those endpoints fail and then either modified or developed new endpoints. But what I would say that we did that was a little bit different than others, I think, and I encourage everyone to consider doing this when you do have endpoint data and some natural history data, if it's older or you haven't had a lot of input, is we had a patient-focused listening session where we met with the FDA, and there were about 50 representatives from CBER, CEDAR, the pediatric division, COA, the rare disease division. I mean, there were so many people there from each area, and they just listened, and we presented a very robust kind of summary of the natural history data and other endpoints that were evaluated in the population retrospectively. And what they met patients, they met 5 patients, and they asked the parents, what is the most important, meaningful change for you and your child? And what was very clear is we had done an internal survey, a social media survey, while we were doing the full disease concept model in a more proper psychometric manner. And the survey supported the disease concept model completely, but the parents' responses completely supported the survey as well. And so what we found was every parent wanted their child to have better communication across the board. That's what they wanted. And so the FDA heard that loud and clear. And when we presented what we had on a communication measures, the Bailey, the Vineland, the communication matrix, their statement to us was, some guy writing on a piece of paper passed it across the table to us and said, this is your communication endpoint. Keep it simple. And then someone sent us an email from CBER connecting us to a team at Duke University saying, you need to help them design this endpoint in communication because there's nothing really that exists out there for this population, and that's the most meaningful to them. And so we worked with Duke to develop this endpoint now called the ORCA, and we developed this for Angelman syndrome. And now the FDA just gave over $2 million to the Duke team to develop the ORCA for 11 other neurodevelopmental disorders, including Rett syndrome. Rett is now working with them as well. But that was the FDA's idea. And so getting the feedback and really having that conversation and that relationship with the FDA is incredibly important. Now, I didn't answer your question because that didn't happen before the NIH natural history study, but things are going to morph along the way. And having the conversation is so important. And what we realize is the FDA is really proud of that measure because they helped to create it. And because of that, they see the value. They see now our kids score in the 50th percentile on a bell curve, and no one's on the floor and no one's on the ceiling. And now in the first clinical trial, that seems to have moved significantly, like by a standard deviation. And so ultimately, the FDA is proud of it. They want it developed for other diseases. They want to work with the teams and the disease, the advocacy groups and the families in order to ensure that important, meaningful things are captured. So keep the conversation and the dialogue going for sure. Yeah, and that's a good way to reach out is through patient advocacy.
Yeah, absolutely. It's a great story. Thank you.
Yeah.
Dominique, just a comment, if you would, on your natural history study, and were you able to connect with the FDA before, or did you have to do it sort of midway?
Yeah, so as Allyson mentioned, the original natural history study was one of the RDCRN consortium studies. And so the FDA was not engaged at that time. When we went through the third cycle of funding, and it became just a Rett and Rett-related disorder consortium, it still did not include the FDA, which I think now, in retrospect, many of the PIs say, well, had we known, or had we thought of it, or had there been, conversations like are happening now, we would have involved the FDA at that point. The funding for that study is actually ending this month. And as a foundation, we are launching a new registry, and we are engaging the FDA to look at the data elements that we're collecting and getting their input on it.
That's great. So but
it is an iterative process, and it will change, and we hope to continue to engage FDA as well as industry, because industry also has expertise in what the regulatory needs are of registries.
Right, absolutely. And I think that's a strong message for everybody to hear, that it is going to change over time. Another question for you, Dominique, was the drug that you reached out to, that initial pharma company, a previously available drug, but you recommended an off-brand usage?
Oh, great question. It was not a, repurposing. So it wasn't a drug that was already FDA approved. It was a drug that was in clinical trials for other conditions. It was for traumatic brain injury. But because of some of the mechanism made sense for Rett syndrome, the conversation was initiated to say, why don't we look at this preclinically in Rett syndrome, in the Rett syndrome mouse model, to see, does it actually demonstrate efficacy? And then that launched us into clinical trials, which now is in a phase 3 clinical trial.
That's great. So another question, Geraldine Bliss, for Allyson and Dominique. Geraldine writes, your kids like mine have disorders that typically don't get diagnosed until there are some obvious signs of developmental problems or regression, or comorbid health conditions. It seems like gene-targeted treatments have more potential if we can treat early, even before the onset of developmental problems. What are your ideas about trying to get kids diagnosed at earlier ages? Allyson, you want to take it first?
Sure. So I think there's a lot of depth to that onion that we can peel back. But for me, I think that the dogma is the earlier the better. And certainly the earlier diagnosis, the earlier intervention, the potentially better outcome you would have. And some of the animal models would support that. And there's contradiction, because there's other animal models that support that you can see full phenotypic rescue in adult models as well, at least for Angelman syndrome. So I think it depends. Do you have a degenerative disorder or a non-degenerative disorder? What is the biology? Do you have a anatomical defect, or is it just a not structural, or is it just biochemical? So when you're talking about something like cerebral palsy and more of a hypoxic event, that's very different than if you're talking about some type of genetic disorder. And I know the question was more in a genetic disorder. So I do believe the earlier you intervene, the better chances you probably have theoretically, and there's dogma to support that. But I do believe there's new data to say that you can see significant changes over time, and the plasticity of the brain is pretty tremendous. And I think a lot of the data that has more recently come out in Fragile X, that was just published in the adult population, where they had a significant improvement in communication abilities in the 18 to 40-year-old group of Fragile X, and in the Angelman syndrome interim data that just came out that showed that individuals with Angelman that are completely nonverbal were speaking words within a 3-month period of time between the ages of 5 and 17. I think that there's a lot of data to contradict that the mouse does not predict the human brain. With that said, getting earlier diagnosis is becoming easier because of the panels, and thank goodness to Safari and all the work that they did in the autism spectrum disorders in terms of understanding the gene association with autism spectrum disorders. With the encephalopathy panels, with the intellectual disability panels, a lot of those panels are really being tested earlier and earlier, which is getting earlier and earlier diagnosis. So we see our population getting diagnosed way earlier now than they did 10 years ago. But yes, if we can have a newborn screen, that would be the ideal situation, except unfortunately, the way the qualification is for the newborn screen it's really the chicken and the egg. And it's really, frankly, quite a disturbing circumstance on how you get on the newborn screen. You have to have an approved drug. You have to have the test that works in order to be considered on a newborn screen that is state dependent. But if you need an approved drug for efficacy based on younger kids that aren't yet diagnosed, then how do you diagnose them if they're not on the newborn screen? So it's this terrible cycle that really needs reevaluation. And I don't need to be the person standing on the podium about this, because there are many other people that are fighting this battle. But it's a battle to be fought, because the earlier you diagnose these kids, not only for therapeutics, but for therapy interventions, for speech therapy and PT and OT, and I mean, for so many reasons, the earlier you're diagnosed is likely better. But how you get that newborn test without it being mandated on a newborn screen is really difficult.
Yeah. Dominique, anything you'd like to add?
Yeah, I'll just add, so for some disorders like Rett syndrome, having a mutation on that particular gene does not actually guarantee that you'll have the disorder. So while the majority of individuals that we know of that have a mutation on MECP2 do have Rett syndrome or a Rett-related disorder, an MECP2-related disorder, there are individuals that have a mutation that make it into adulthood and are married and have a job and may have other medical issues, but they clearly do not have Rett syndrome. And so there's the ethical question of what do you do with knowing that there's a mutation on this gene and not knowing if you're actually going to develop the disorder. And so I think that's one of the challenges with Rett syndrome and maybe some other similar conditions. But otherwise, I agree with Allyson in that it would be great to be able to have earlier diagnosis, especially as more curative approaches are being investigated.
And I would just make sure that everyone takes a look at the chat. A couple of links have been put in there. So good resources for everyone to take a look at. Natalie, a question for you. You talked about to help get an earlier diagnosis, you used implementation science to address that gap between evidence-based practices and the clinical implementation. And then you rolled it out locally and then nationally. Can you talk a little bit more about that and maybe start with a quick definition of implementation science?
Thanks. Yeah, so implementation science is an entire group of scientific methods that encompass some more known ones, such as quality improvement science, but also research on knowledge translation, dissemination, and other techniques. So not just quality improvement. There are lots of different ways of doing this and lots of really great public health schools that have degrees in it. And the NIH has been really supportive of initiatives, including clinical trials that incorporate a strong implementation element so that translation is built in to the studies so that there is a conduit for early and rapid translation of research into practice. What we did is we had to start from scratch from the theory, which is all these massive amounts of studies that had been done throughout the world that showed us through systematic reviews or pilot studies that certain tests were the best test to detect even very early on. And then we had to say, all right, some of these tests are really only for researchers. How do we develop workshops? How do we develop tools to teach them in practice? So some of it was creating from new tools. And we started with those. We tested them out locally and then expanded them. Basically, it's education and it's testing whether your education sticks, whether it's sustainable, and then working progressively at a more regional level. So if you start in one clinic, it might mean going to every clinic in a hospital, saying, if we're going to detect cerebral palsy, we can't just do it in a neurology clinic. We also have to do it in a NICU follow-up program. We have to be able to do it in outpatient therapy services, everywhere, or even in the pediatric community, everywhere where it actually occurs. So there's that. And there are different phases of implementation science. I showed you the national one. The national one required a network to scale up. And that was much more difficult. The first thing you do is you make sure everyone's on the same page, is implementing exactly the same thing at the same time. It's not romantic, it's not glamorous. It really takes advantage of techniques that are used in Lean Six Sigma business processes. So the business world has a lot to teach us about how we approach early detection, actually. And so I would make sure that people understand that a lot of these things don't require a PhD or an MD. They actually require you looking at successful models of either knowledge translation or process improvements or just implementation into healthcare systems. And so we used a lot of those tools, and now we're at the stage where we're starting to work at the policy level. So we're working with the American Academy of Pediatrics, right, to make statements so that we've shown that this process can be scaled up pragmatically. And now we need to work with the academic bodies that rule policy on this, the CDC as well. And we make this into policy statements, so then the scale up can be at a much higher level. And so I guess that's a long answer to your question, so I apologize.
Yeah.
But
No, it's helpful, I think, yeah. I think, again, we're all learning from one another, right? So it's a difficult topic to approach, right? You've got to get everybody rowing in the same direction. So, Allyson, you talked a bit about the researchers having weekly calls. That may have happened before you started, but how did that happen? I mean, how did they get them to do that? Because, I think a lot of us are still working with researchers who tend to work in silos, although it's better than it was before, but that still is some of the reality that we're facing.
I think it happened based off of passion and the desire to see collaboration. So it started with the founder of FAST, who is Paula Evans, and her relationship with one of the researchers at the University of South Florida named Ed Weaver. And he had published the gene therapy in a mouse model of Angelman syndrome. And they started, having a relationship and really understanding what the potential was for Angelman syndrome research. And he had come out of a different lab and had moved to University of South Florida. And so he was able to just recruit some colleagues in his other lab that were, focused on, EEG and electrophysiology, and other people that were more focused on creating animal models, and other people in epigenetics. And it really these were all people that had either worked a little bit in Angelman syndrome or were interested in working in Angelman more as a colleague to colleague discussion. And that ultimately morphed into, let's create a team, a research environment that we can fund in totality, that you guys can leverage each other's expertise and accelerate each other's publications and funding. And it was very, very well received. And again, this was before my time. That the chief science officer before my time was Becky Burdine, and she was integral in developing and designing this as well. And I think it was just, we were lucky to have really good nature researchers who wanted to see Angelman syndrome reach another level. And there was a lot of thought that Angelman is one of the more common rare diseases. To be perfectly honest, it's probably one of the more simpler ones to address, in that there's 2 or 3 approaches that you can take, generally replacement or paternal activation, compared to something like Rett syndrome, where you have the X chromosome and Y chromosome. Things are a bit more complicated in many of these other haploinsufficiencies or sex-linked disorders, and you don't have that with something like Angelman, and it's non-degenerative, and you can just activate a silent gene that's sitting there, and it's neuronal only. So there were so many reasons that people wanted to see it be successful, but for whatever reason, NIH wasn't funding it as they were funding many other neurodevelopmental disorders. And so the foundation just kicked in and said, we're going to fund it. And the researchers were like, let's do it. And they all just got on board. And it was a beautiful thing. When I came in, it was relatively well established. And we brought in a couple more people in my time there. And they've just been amazing friends and colleagues, and very pre-competitive and very supportive of one another. And that was very much responsible for a lot of the progress.
Yeah, that's terrific. I know some organizations have had to build it into their funding terms and conditions that, that you receive funding based on your willingness to collaborate with others, which I think is also a strategy.
And we also have if we fund you, we have a translational research symposium that we have every year, and all of our scientists that we fund have to present at it, and they have to present their data, even if it's not published yet, even if it's not patent ready yet. Ultimately, it's a closed room in which there's no pictures allowed, only scientist to scientist. Everything in there is confidential. There's rules and regulations for you to be allowed in the room, and the room is there for people to give you feedback and for people to hear what's going on, so we don't duplicate efforts. One foundation doesn't fund the same thing the other foundation is funding because everyone's private, and no one publishes negative data, which I find abysmal. And so what happens is everyone wastes money on redoing data that they don't know is already negative. So here is a space to present negative data, present collaborative data, present really exciting early information so people can work off of that together. And it's a very respectful environment, and no one's broken the rules yet. So it's been a really nice collaboration. And we have 150 people in the room, and it's really nice.
That's terrific. Congratulations on that.
It's not me, it's them.
Well, sadly, our time has come to an end. I just want to again thank each of you for sharing your stories with us, inspiring us. Dominique talked about the importance of small groups. I think it was Margaret Mead who said, "Never doubt that a small group of committed citizens can change the world. Indeed is the only thing that ever has." So we are inspired, and it's a nice way to end the forum. And I'll turn it over to Sam or Walter to wrap it up. Thank you again for your wonderful presentations.
Thank you so much, Janet. Thank you for stepping in kind of at the 11th hour and leading such a brilliant conversation with these speakers. You all were fantastic. It was really inspiring to hear about your success stories and to hear your thoughts during that live Q&A we just completed. So with this, that is the last session of the 2021 forum. And so I want to pass the mic back to our NINDS director, Walter Korschitz, to hear his final thoughts and see if he has any last questions for our community.